Hi all, we are looking at question number 20 from GATE Biotechnology 2015 paper. So the question is, which one of the following amino acids has the highest probability to be found on the surface of a typical globular protein in aqueous environment? The options are A. alanine, B. valine, C. arginine, and D. isoleucine. To answer this question, let's learn about the properties of amino acids. 20 different naturally occurring amino acids make up polypeptides and proteins. Polypeptides may be di, tri, and so on up to 10 amino acids, while proteins are longer than 10 amino acids and usually weigh above 10 kilodaltons. The structure and function of proteins depends on the order of amino acids that make them up. For example, they form structural components such as cartilage, muscles, hair, and nails. They can function as enzymes to regulate biological reactions such as digestion and cellular metabolism, like hemoglobin and myoglobin transport oxygen in the blood. Here are just more examples of proteins and their functions. For example, collagen is a structural protein that provides structural components. Keratin is another such protein that provides structure to the hair, skin, wool, and nails. And myosin is a contractile protein that makes the muscles move. And like we just saw, hemoglobin helps to transport oxygen, and lipoproteins transport lipids. So they are involved in transport. And casein is a protein that stores milk, so it's a storage protein. And hormones like insulin regulate blood glucose level, and growth hormones regulate body growth. So there are also enzymes that catalyze biochemical reactions in the cells. For example, sucrase catalyzes the hydrolysis of sucrose, and trypsin catalyzes the hydrolysis of proteins. And there are also proteins involved in protection. So they provide protection by recognizing and destroying foreign substances. For example, immunoglobulins stimulate immune responses. So these are just examples of different categories of proteins functionally. So amino acids, which are the building blocks of proteins, have a central carbon atom, and this is called the alpha carbon. It is bonded by two functional groups. One, the ammonium group, which is NH3, and a carboxylate group, which is COO minus. So it has a hydrogen atom and a side chain group called R. So the charged ammonium and carboxyl groups cancel out in charge and make the amino acid neutral. This neutral amino acid is called a zwitterion, and the pH at which it occurs is called its isoelectric point. So here's an example of the structure of zwitterions of amino acids like serine and aspartate. So further, based on what R group is present, the amino acids are further classified as aliphatic, aromatic, sulfur containing, and based on their water solubility, they're classified as polar and non-polar. So the polar amino acids are hydrophilic and the non-polar amino acids are hydrophobic. Here's a summary of amino acids and their interaction with water. So there are 9 amino acids that are non-polar in nature. They are also hydrophobic, that is, they're water repellent. So there are groups, they consist of non-polar groups like aromatic groups. And there are 6 polar neutral amino acids, which means they are hydrophilic, but they are chargeless. They are just neutral in charge. So in their R group, they contain O and S atoms. And next one is the polar acidic type of amino acids. So there are 2 polar acidic amino acids. They contain carboxylate groups, that is the COO minus group, and they are negatively charged. So these are hydrophilic as well. And there are 3 polar basic amino acids. So they contain ammonium groups, that is the NH3 plus, and they are positively charged, and they are hydrophilic. So let's look at what these amino acids are. So the non-polar amino acids are hydrophobic, like we just saw, and they usually have a hydrogen alkyl or aromatic side chains, and there are 9 polar amino acids: glycine, alanine, valine, leucine, isoleucine, phenylalanine, methionine, proline, and tryptophan. So amino acids are represented by 3-letter and 1-letter codes. So for example, glycine The codes, the 3 letter code is GLY and the 1 letter code is G. So similarly for all the amino acids we have these 1 letter and 3 letter codes. So you could just easily remember the non-polar amino acids by this acronym GVAMP WILF. So yeah, this is just for you to remember it. It's nothing technical. It's just so that you easily remember uh, the amino acids. So the next category is the polar amino acids, and among them we'll now look at the polar neutral uncharged amino acids. So an amino acid is polar neutral when the R group is either an alcohol, that is like it has an OH group, or a thiol, that is it has an SH group, that is like a sulfur containing analog of alcohol OH, or an amide. So amide is a carboxyl group that's attached to an amine group. So we have 6 amino acids that are polar neutral, and they are serine, threonine, asparagine, glutamine, tyrosine, and cysteine. So you could remember these by the acronym QUISINS. So if you might know, there's an organization called Queer College Youth Network Triad. So yeah, you could just easily remember all these amino acids which are polar uncharged with this acronym. So these denote the 1 letter codes of each of these amino acids. So next, among the polar or hydrophilic amino acids, there are 2 acidic amino acids. So they contain a carboxyl group, that is COO minus, and they are negatively charged at physiological pH. So these carboxyl groups also function as nucleophiles in some enzymatic reactions. So the 2 polar acidic amino acids are aspartate and glutamate. So they are represented by D and E, or ASP and GLU, which are the 3 letter codes. Then there are 3 polar basic amino acids. So again, they're hydrophilic and have nitrogenous bases in their R group. So they are positively charged at physiological pH. So the 3 polar basic amino acids are histidine, which has an imidazole ring, lysine, and then arginine. It has a guanidium ion. So this part is the guanidium ion. So these 3 are the polar basic amino acids. So of these, arginine is the most basic amino acid. We also saw that based on their side chain group, that is the R group, they're classified as aromatic amino acids, aliphatic amino acids, or sulfur containing amino acids. So like the name suggests, aromatic amino acids has aromatic rings in their R groups. So phenylalanine, tyrosine, and tryptophan are the 3 aromatic amino acids. And then there are aliphatic amino acids, that is they have alkane groups in their side chain. So the aromatic and aliphatic amino acids are all hydrophobic. So these are the aliphatic amino acids. And proline is an amino acid. It's not an amino acid, but it's an amino acid because it has an imino group instead of an amino group. That is, it has NH instead of NH2. And then there are sulfur containing amino acids. They have sulfur atoms in their side chain. So methionine, which is encoded by the start codon, has a sulfur, and the other sulfur containing amino acid is cysteine. Further, all these 20 amino acids that we discussed are standard amino acids. So these amino acids, they are used to form proteins. They are recognized by ribozyme auto aminoacylation systems, that is the translation system. And apart from these, then there are non-standard or non-protein amino acids. So these are not required to make proteins, but they are the derivatives of the standard amino acids, and they have a role in metabolism, and they act as intermediates in several metabolic processes. So some examples are hydroxyproline, alpha aminobutyric, ornithine, citrulline, beta-alanine, hydroxylysine, carboxyglutamate, and so on. So further, they could also be classified based on their source. For example, only 11 can be synthesized in our body, and the rest 9 must be obtained from proteins in the diet. So these are called essential amino acids, and the ones that are synthesized in the body are called non-essential amino acids because we don't need to acquire them from external sources. So, animal proteins from egg, milk, meat, and fish. So, all of these are complete proteins and contain all the essential amino acids. So, whereas proteins from plants such as grains, beans, and nuts are deficient in one or more of the essential amino acids, and hence they are incomplete proteins. For example, if you look at this chart, so we have legumes. So legumes, they do not have a methionine and tryptophan amino acids, while they have isoleucine and lysine. So whereas grains, they don't have isoleucine and lysine, but they have methionine and tryptophan. So that's why when you eat these two together, you'll acquire all the amino acids. So that's called like balanced diet. Especially when you have vegetarian diet, you have to like take have these legumes, grains, and rice, all of these together, so that you get all the essential amino acids. So, the protein structures can be classified as primary, secondary, and tertiary structures. And if the protein has more than one chain, it can have a quaternary structure. So, like looking at the primary structure, the simple linear sequence of the amino acids in the peptide chain, along with the disulfide bridges formed between the sulfur atoms in amino acids, is the primary structure. The next level of protein structure is the secondary structure. So it refers to local folded structures that form within a polypeptide due to interaction between atoms of the backbone. So the backbone just refers to the polypeptide chain apart from the R groups. So all we mean here is that secondary structure does not involve R group atoms. So the most common types of secondary structures are the alpha helix and the beta pleated sheet. So both structures are held in shape by hydrogen bonds which form between the carbonyl O of one amino acid and the amino H of another. So many hydrogen bonds are sufficient to define this shape. The next level of structure is the tertiary structure of proteins. So these arise from weaker attractive forces like non-polar dispersion forces between the hydrophobic parts of the same chain that are widely separated in the primary structure but close in space. So that is the amino acids that are even far apart in the primary chain. They are brought close together in the tertiary structure because of these weaker attractive forces, non-bonds. So tertiary structure is intramolecular. That is, these bonds form between, like, they don't form between different molecules, so they just form within the same molecule. So this results in chain twisting and folding. So there are two major tertiary structures like braids and globes, like globulins. So for example, collagen, it's a fibrous protein and it's a precursor of gelatin. It has a triple helix structure and it has some elasticity because of the interchain interactions. So collagen is made of 3 peptide chains, and there are bonds between these 3 peptide chains, and that contributes to the elasticity. And the other example, we have hemoglobins and albumins. So these are globular proteins. So globular proteins are named for their approximately spherical shapes, and these are the most abundant proteins in nature. So these globular proteins exist in an enormous variety of three-dimensional structures. So have you ever wondered why egg whites go from clear to opaque when you fry an egg? So egg whites contain large amounts of proteins called albumins, and the albumins normally have a specific 3D shape due to the bonds formed between different amino acids in the proteins. So heating causes these bonds to break and exposes the hydrophobic, that is the water hating amino acids, which is usually kept on the inside of the protein. So these hydrophobic amino acids, they try to get away from the water surrounding them in the egg white. So they stick to one another, forming a protein network that gives the egg white structure while turning it white and opaque. So the structure and integrity of the proteins depend on how the amino acids are located in the tertiary structure. So ideally in an aqueous environment, the water loving, that is the hydrophilic amino acids, are faced outside, while the hydrophobic amino acids are faced towards the inside. So now coming back to our question, which one of the following amino acids has the highest probability to be found on the inner surface of a typical globular protein in an aqueous environment? So ideally, in an aqueous environment, on the outside, on the surface, there has to be polar amino acids. So out of the given options, if you remember the acronym GVAMP-WILF that I suggested you to keep in mind to remember the non-polar amino acids. So according to that, like option A, B, and D, A, V, and I. So all these are non-polar amino acids. So that leaves us with arginine, whose one letter code is R. So this is a polar amino acid. So that answers our question.